Good afternoon. My name is Anthony Jolly. My research is a meta-analysis study focusing on the biological implications of age and gender on lung carcinomas. So, what is lung cancer? Well, cancer is known as the uncontrollable growth of cells in the body. So, lung cancer is the uncontrollable growth of cells in the lungs. It affects the pleural membranes in the lungs by either causing a buildup of fluid or blocking major airways, both of which induce shortness of breath and difficulty breathing. Lung cancer is the second most diagnosed form of cancer. It's responsible for about 1.6 million deaths each year. So, why is this important? Well, being able to identify factors that increase the risk of getting a disease is essential to diagnosing and treating it. Smoking has been evidently shown to increase the risk of developing lung cancer in both males and females. However, sex, age, diet, and even region have not been fully explored as to whether they play a role on the prevalence of lung cancer in individuals. By looking at these factors, we could also identify different biomarkers associated with cancer for these different groups. So, the goal of this meta-analysis study was to identify individuals that are at high risk of developing lung cancer and to identify possible biomarkers associated with lung cancer. So, for our methods, we managed to do the data curation and the dataset preprocessing. So, for the data curation, I looked on the NCBI PubMed database and looked for anything that matched the criteria of lung cancer or lung carcinoma, Homo sapiens, and expression profiling by array. If it didn't match, then we'd exclude it. If it did, then we'd see if it reported age, gender, and smoking history. If not, then it would be excluded, and if it did, then we would download the raw expression data. This step took a really long time to do, and it was very annoying. So for the dataset preprocessing step, we would take the downloaded raw expression data and we would annotate it so we know which genes were which. And then after that, we'd run a background correction and a normalization just to make sure that everything was within the same scale. Then we do a batch effect correction. This was to eliminate any errors that might have been given from the machines and not necessarily the computational analysis. And then after that, we would run a linear model analysis. We didn't get to do the linear model analysis. We only managed to do the pre-processing, but the pre-processing step took so long because we kept running into issues with the normalization and the background correction. It was either the data sets were wrong, it was either the program could it was either that the program that we were using could not read it, or we just had to use something completely different because it ended up being that they were annotated differently. So that took us a really long time to figure out. Okay, so from our data creation step, we managed to find 6 data sets that met our criteria for age, gender, and smoking history. On the left, this is a chart showing the amount of diseased females and diseased males. There's a total of 381 females with lung cancer and a total of 316 males with lung cancer. This is important because we have more females with lung cancer than we do males with lung cancer. So, um, So we might expect to see a higher expression profile in females than males. However, this would be biased because we have more female samples than we do male samples, which is something that we're going to need to correct for downstream. In total, we have 934 samples, with 131 of them being female controls and 106 of them being male controls. So, what next? Well, in the future, we plan to run a linear model analysis to see if there's an actual relationship between these factors and lung cancer. Then we plan to do a p-value adjustment so we can extract the genes that showed a significant change in either upregulation or downregulation. Then we plan to do a two-key honest significant difference test so that we can see it, basically to see if these results are actually answer our question or are significant to our question. And then we plan to graph the up-regulated and down-regulated genes by factor, and then do a geo enrichment analysis to see what pathways and how these genes are interacting with each other to cause this upregulation or downregulation. So I'd like to thank the UVI ECS program for giving me the opportunity to do this research, and the NIH RISE program for providing the money so that we can actually do all of this. But most importantly, I would love to thank my mentor, Dr. Levita Rogers, for helping me through this, because at the beginning of all of this, I did not know what in the world I was doing, and I was completely lost. So if it was not for her, I would not be able to talk today at all, because I would not know what I'm talking about. And that is my presentation. Thank you.